Thank you so much, Donna. And John, it's delightful to be here with you to lead this conversation today.
Thanks for having me. It's a pleasure.
Well, John, you earned your PhD in molecular biology at the University of Chicago, and then you went on to work at several biopharmaceutical companies before becoming the founding CEO at Alnylam. How do you think about the intersection between the basic research that goes on, largely federally funded, and the use of the fruits of that research by the private sector, and how this a wonderful symbiosis works together to accelerate biomedical progress?
Well, Susan, both the public sector funded research and the private sector, to use your words, a symbiotic relationship without a doubt. They both play a major role. Obviously, the public sector funding of research is so critical to the foundation or the discovery of new ideas that can lead to tomorrow's medicines. And then the private sector funding can come in to really provide the funding for the translational work that's needed to bring innovative medicines to market. And I can give you a really good example with Alnylam and RNA interference. The NIH funded about $10 million of research in the labs of Andy Fire, Craig Mello, and some other investigators in the public sector. Obviously, Fire and Mello received the Nobel Prize, which they certainly deserve, for the discovery of RNA interference in C. elegans. In terms of what Alnylam did, is we invested about $6 billion to bring 4 RNAi therapeutics to market over a 20-year period. So the work that we do as a private sector company and the work that the public sector does are so symbiotic with each other, but the aspects of the funding environment that's needed to do one versus the other can be very different. But that's a great example in our history for how the two work together hand in hand.
Now, you mentioned RNA interference, the core of Alnylam's work. Give us a little science lesson today. What exactly is that, and why is it viewed as really one of the most promising frontiers in biology today?
Well, Susan, RNAi is a revolutionary discovery in biology that really underscores a natural process for the regulation of gene expression. It's akin to transcription factors in DNA or the proteasome and proteins. The ability of harnessing this natural pathway to silence specific disease-causing genes targeting the encoded messenger RNA really defines a new way to treat disease. There's so many advantages of targeting RNA as compared to other approaches where you might target DNA as a way of advancing genetic medicines. For example, you can achieve a reversible effect and avoid the uncertainties that are inherent in DNA targeting therapies, let alone all the bioethical considerations that go into targeting DNA. And as for today, RNAi-based medicines are now a reality. There are 4 approved medicines that have been brought through clinical trials to approvals in countries around the world. And these medicines range from medicines for inherited diseases to prevalent diseases like hypercholesterolemia. So there's been a lot of progress on how this basic discovery of RNA interference has now really opened up the door for new medicines to be developed.
In terms of the awareness of the public about this kind of research, obviously a different area that still has the word RNA in it is the mRNA vaccines that have been developed and are now in widespread use in the COVID pandemic. Has that awareness advantaged you in any way in terms of the public understanding of the importance, at least of RNA generally?
Well, if you had to find a silver lining of the pandemic, at least for our science and technology, it certainly was the remarkable scientific advance of mRNA vaccines and what I like to refer to as the democratization of RNA-based medicines. And obviously, that's had an enormously positive impact on the public understanding of the types of medicines that we make at Alnylam that are really using RNA, no different than the RNA that billions of people have received as part of the vaccination against COVID-19. So there really has been a benefit for RNAi-based medicines because of our cousin with mRNA and how they have, of course, advanced and really taken us out of this pandemic, or almost out of this pandemic.
So the therapies that you develop, they are, the general pool of therapies that Alnylam develops work with several different delivery platforms, as I understand it. What's the difference among those platforms, and why should we understand that there are these different platforms to deliver the therapies?
Yeah. Well, listen, one of the major challenges of making drugs out of small interfering RNAs, which are the molecules that mediate RNAi, was achieving delivery of these large, charged, and highly degradable RNA snippets into the cell cytoplasm where they can harness the endogenous RNAi pathway to achieve target gene silencing. And through the work that we did, and we did this collaboratively with other academic groups and other companies, we found two basic approaches for the delivery of small interfering RNAs to hepatocytes in the liver. And the first of those was lipid nanoparticles, which basically encapsulate the RNA and release the RNA into the cytoplasm after bursting open the endosomes. And LNPs were used in the development of a medicine called Onpattro, which is the first RNAi therapeutic ever approved for a disease called hereditary ATTR amyloidosis in patients with polyneuropathy. The second approach was to attach a small molecule ligand to the siRNA molecule and to achieve a receptor-mediated uptake of the siRNA into the cytoplasm. And this conjugate-based approach used a sugar molecule, that is the ligand, to achieve uptake by a sugar receptor on the hepatocyte cell surface. Now, interestingly, Alnylam's work on both LNPs and conjugates has found applications in other fields. We just talked a minute ago about mRNA, and actually the work that we did on lipid nanoparticles has been used for the advancement of mRNA vaccines. And as it relates to conjugate work, the work that we've done has actually been used to deliver other oligonucleotide therapies like antisense oligos or microRNA therapeutics. So there's been a lot of utility of these delivery technologies in different fields, in addition to how we've used them for RNAi-based therapies.
So Alnylam has, as I understand it now, a total of 11 clinical programs. You mentioned one of them, Onpattro. Most of these really fall into the categories of treatments designed for what we would call orphan diseases, correct?
That's right. That's right.
Which means relatively small populations of people. How long can you keep going? Are there that many orphan diseases left that can be addressed through this pathway, these pathways?
Well, look, there are over 7,000 orphan diseases, and we've tackled 3 of them with approved medicines right now, and many of the programs in our pipeline are looking at others as well. So it is a large number of diseases that we could tackle with this. Now, of course, what really is critical is where we can get good delivery of these small interfering RNA molecules, and we've done this with hepatocytes, and we've been able to tackle inherited diseases with genes expressed in the liver. But more recently, we've made progress in delivering RNAi therapeutics in the CNS. And so that opens up the opportunity of advancing medicines for many neurodegenerative diseases that are caused by overproduction of disease-causing proteins. So RNAi, of course, is really well suited to that application. So I do think there's a bright future for many RNAi therapeutic opportunities for a much larger number of orphan diseases. But there's also an opportunity, which we're increasingly exploring, for advancing RNAi-based medicines for more prevalent diseases, including diseases like hypertension, which is the leading cause of CV morbidity and mortality around the world, and also a major public health concern. So it's, of course, largely been in orphan diseases, but we are now opening up the door for RNAi-based therapies in more prevalent diseases as well.
Wow, that could be extraordinary to really tackle some of these very, very prevalent diseases that continue to be the predominant killers of Americans.
Exactly.
What partnerships are you going to need, and what partnerships do you have now with others in the biopharmaceutical arena and even back to basic science to continue to do this work?
Well, listen, on the partnership with other biopharmaceutical companies, we've had a long history of forming partnerships. We've had over 25 partnerships that we formed since 2002 when we got started. A great example of that is the partnership we have with Regeneron, where we're focused on advancing RNAi therapeutics for both CNS and ocular diseases. And it's a great example of taking two like-minded, science-focused companies, Alnylam and Regeneron, and joining forces to advance innovation for diseases of the CNS and the eye. So that's an example on the company side. On the academic side, we've really made a big commitment in human genetics as part of our overall R&D effort. To this end, we have partnerships with the UK Biobank and also with UCLA, a partnership that we formed just last year, which are really aimed at connecting genotype and phenotype in the discovery of novel genetic targets for the future of RNAi therapeutic programs. And that has been a remarkable collaborative effort that we've joined into, which really is going to be the foundation for the future of novel target discovery in the work that we're doing.
What do you think your company needs most from the broader research ecosystem to continue to advance RNAi-based treatments? And what do you see as the greatest opportunities as well as challenges?
Well, listen, there are, of course, many challenges and opportunities, and they tend to be related to each other, as you can imagine. But without a doubt, the greatest challenge opportunity relates to the continued expansion of delivery sciences for RNAi therapeutics. There's been great progress for targeting cells of the liver and also more recently cells of the CNS, but there are still many, many target cells and organs where new innovation is just required. And we really do expect much of this to happen in the academic communities through basic research. For example, if we can conquer delivery of small interfering RNAs to tumors, we could have a powerful platform technology to silence oncogenes and have a new approach for the treatment of many, many cancers. So we could benefit enormously from new delivery platforms discovered in academia, and we would welcome the opportunity as a company to partner with academic leaders in this effort, which we, of course, can't just do internally at Alnylam. We have to really rely on the great efforts of basic research around the country and the world.
Well, John, you've made the case again so powerfully about the need for this symbiosis between basic research and the translational component, and eventually leading into the commercial opportunities that your company has brought to market. I want to thank you so much for joining us today for this terrific conversation.
Thank you, Susan. A real pleasure.